Hi, everybody. Thank you guys so much for being here today. My name is Alexa Tinsley. I am the Director of Community Engagement for Jett Foundation. We have the Edgewise team here today to talk about their novel approach to protect muscle in Duchenne and Becker muscular dystrophy. I am going to give people a few minutes to hop on. Thank you guys. It'll be about like 30 seconds to a minute, but thank you guys for your patience while we dealt with some technical difficulties. But while we were waiting, if you could put your where you're located and your child's age in the chat, that would be wonderful. And we will get started in just a minute. All righty. Let's see. All right, we'll go ahead and get started. Again, my name is Alexa Tinsley. I am the Director of Community Engagement here at Jett Foundation. Again, thank you all for joining our community webinar series. We're so glad that you're here. We are welcoming Edgewise and their team to discuss their novel approach to protect muscle in Duchenne and Becker muscular dystrophy. And we are so grateful to partners like Edgewise for making this happen and us being able to bring the education and the knowledge on these, upcoming trials and treatments into your home. So I do want to welcome Abby Donovan, or Joanne Donovan. She is the Chief Medical Officer for Edgewise, and Abby Bronson, who is the Vice President of Patient Advocacy. We are also hoping to see Dr. Alan Russell, who is the co-founder and Chief Scientific Officer at Edgewise. He's having a little bit of trouble signing on, but he's trying, so he should be joining shortly. But we're very excited to have them join us. Before we get started, I do want to mention a few things. Today's webinar will be about 30 minutes long. We will have a live Q&A at the end of the presentation. You do have a Q&A box at the bottom right of your screen, so you should be able to type questions in there. We will make sure that we do our best to get to them in a timely manner. It is being recorded, so if for any reason you have to log off or you're not able to make the whole thing, or there's someone that you know that wanted to be here today but wasn't able to because of the timing, we will have this recorded on our website and our YouTube for you to watch later, and you can always pass along that link to others as well. So we will be looking at the chat and do our best to get all of the questions if questions do come in through the chat, but our main portal will be through the Q&A. So if you could try to use that, that would be very wonderful. And if there are any questions at the end, again, we will do our best to get to them. If there are any questions that we don't get to, we will make sure that it gets to the Edgewise team. Hi, Alan. Alan is here. So this is Dr. Alan Russell. He is the co-founder and Chief Scientific Officer at Edgewise. So he will be presenting as well today. So thank you, Alan, for being here. I'm sorry that we had a little trouble, but you're here and that's what matters. So with that, I will hand it over to Abby. Thank you, guys.
Thank you. Hi, everybody. And first of all, thanks a lot to Jett Foundation for organizing this, because this is how we get our messages out, and we're really pleased to be here. So you can go to the next slide. There. So this is our forward-looking statement. We're going to be talking about things that potentially might happen in the future. We're a publicly traded company, and probably the most important thing to remember is that EDG-5506, which is the investigational compound we're talking about today, it has not been approved in any territory. So next slide. So we're going to talk to you today about, first of all, a little introduction to Edgewise and what patient advocacy is like here. And then our approach, and Alan Russell will take you through, Dr. Russell will take you through some of the science, and then our clinical development program and a little bit about some of the original study that we did in Becker, and then what we have planned. So you can go to the next slide. So Edgewise was founded in 2017, and its vision and mission is to develop novel therapies for muscular dystrophy, including Becker and Duchenne. And we really wanted to develop and design investigational compounds that had broad benefit. They could be used alone or in combination. We realized that, having a chronic disease is hard. So we wanted to make sure that they were convenient to administer. And that is what we wake up every day trying to do. So we designed this compound EDG-5506, and that's what we're discussing today. So you can go to the next slide. So before I turn it over to Alan to talk about the science, I wanted to take a few moments just to talk about what patient advocacy is like at Edgewise. And truly, patients are at the core of everything we do, and your voices and listening to you really does matter. And so I think about what patient advocacy does sort of in two buckets. And the first two bullets is sort of the first bucket. And that's really listening to your voices and taking those insights and your experiences. And we use that information. We don't just listen to you, we actually use it to help improve our trial design. We look at the endpoints that we're using. And we also use it to establish something that we call the therapeutic context for risk-benefit decisions. And what that means is, like every sponsor, we do clinical trials. We collect a lot of data on the safety and the efficacy of an investigational therapy. And then we bring it to the FDA and they make a risk-benefit decision. But it's very hard without understanding what exactly is it like to live with that disease? What are their symptoms? What's the disease burden? What's the unmet need? So that's the context from which all those risk-benefit decisions are being made. And we can only get that from you, the patients. And then the second bucket of things that we do are things like this. Like we work with the advocacy organizations, we provide information to you, we work on community-wide projects, collaborations, coalitions, all trying to move research and accelerate clinical trials. So that we can create better awareness and really help the community move forward in all different ways. So next slide. I'm now going to turn it over to Alan Russell, who can talk about our approach.
Thank you, Abby, and thank you, Alexa and Julie and the Jett Foundation for giving us this opportunity to talk about some new biology and how this compound 5506 might help if you have Duchenne or Becker muscular dystrophy, for that matter. So what I'll tell you is a story about muscle fibers, how contraction of muscle fibers causes them to become damaged and degrade when you have compromised dystrophin, and then how we've come about this novel therapy that's in development right now. So as you can see here, we've got 3 bullets. Some muscle fibers are more susceptible to injury than others. You often hear talk of fast and slow skeletal muscle fibers, particularly if you're a sports fan. It turns out that fast muscle fibers, which makes up about half of your body, are much more susceptible to injury than slow ones. And that's particularly the case if you don't have dystrophin. We've made an investigational therapy. It's called EDG or EDG-5506, and that's designed to protect those susceptible muscle fibers. I'll tell you how it does that, and I'll show you a little bit of data to kind of illustrate how it works. Now, this is a mechanical fix. It's not a genetic fix. So it works irregardless of what the background mutation is. The therapy is really designed to be as broadly applicable as possible. So pretty much potentially useful for all sorts of people. And the final bit is we have done a lot of preclinical work in models of muscular dystrophy. And the common theme amongst those studies is that the compound is protective. And then in long-term studies, you see things like changes in fibrosis and fat incorporation, which is really what we want to do in the clinic. Next slide, Abby. What I'll show you is dystrophin in motion. On the left-hand side is a figure you're used to seeing. Dystrophin is this long protein that connects the inside and the outside of the muscle, right? But of course, muscle contracts, and that's what you're seeing on the right-hand side. And this is how people like to think of muscle contracting. The dystrophin-containing complex are those green stripes. So they're very regular. They run up and down the fibers, and they actually connect the fibers together. Now, fibers don't always contract together. Sometimes one contracts and others don't. And in this case, you can see that central fiber is contracting, and the green stripes are helping support that single fiber by connecting it to the other ones. Now, of course, if you don't have dystrophin, the complex falls apart. The body tries to upregulate elements of proteins to try and make do, but it doesn't work very well. And the end product really is that when individual fibers contract, they now have no support from their surrounding fibers. And that leads to an increased chance of stress. You know, if you're all alone, there's just a higher chance of getting stress. And of course, when you get stress, you get membrane stress, and that causes initiation of pathways that tell the muscle fiber that it needs to go away. The body thinks it's not functioning properly. So what it does is it prunes those fibers out by activating proteolytic enzymes to chop that fiber up, and leave essentially an empty tube that the body can grow a new fiber back into, right, using stem cells. Happens to us when we go and work out. If you do eccentric exercise, that's lengthening exercise, you're essentially giving yourself controlled injury. And by doing that, you can grow your muscles. So it's a perfectly natural thing. But of course, if you have Duchenne or Becker, then that rate of turnover is so much higher. The body has trouble keeping up with that. And that eventually leads to the inflammation and the fibrosis that limits function. So it's not actually the absence of dystrophin that gives you a deficit in function. It's actually all of this damage that occurs because you don't have dystrophin that really compromises the ability of the muscle to do what you need it to do. Next slide. You go to the next one. Now, here's everything all at once. So this is essentially a real life muscle contracting in a bath. These are from mice, the MDX mouse model of muscular dystrophy, the Duchenne model. And on the left-hand side, just keep your eye on that one. That's the control muscle that is contracting without any addition of any compound. So what you'll see is essentially that video I showed you before, but now in a real muscle. So you can see things moving around when they shouldn't be. As this muscle is contracting, it's becoming stressed. These proteolytic enzymes are snipping up the fibers, and then you see them retracting, and then you see the formation of these kind of blobs throughout the muscle fiber. And that's essentially these long fibers that have been crunched down to a little ball ready to be disposed of. Now, on the right-hand side, we've treated the muscle for 1 hour with the compound 5506. It's a small molecule, much the same as aspirin or Tylenol or anything else. It's orally available. In this case, we've just put it into the bath for 1 hour. And what that's done is it's changed how the muscle contracts. So 5506 targets the myosin in these susceptible fast fibers, and it changes how the myosin works. Myosin is the protein that essentially controls contraction of fibers. And what it's done is it's applied a speed limiter on that muscle fiber so that it's uncoupled the ability of the muscle fiber to injure itself. So even though the thing is contracting, by applying this myosin inhibitor, 5506, what we've done is we've limited the amount of stress that the muscle fiber can put upon itself. This is not enough of a change of function to really notice anything, but it is a change of function enough to uncouple this stress such that the muscle can now contract normally, even though it doesn't have dystrophin, without breaking apart. So if we can do this, of course, in muscles in kids or adults with Becker or Duchenne, that would profoundly change how people move around. The muscles can contract without that injury response. And that's what we really want to test in the clinic. Go to the next one. Now, of course, you may well be familiar with creatine kinase. It's often the first signal to kind of diagnose or certainly get on the pathway for diagnosis. Now, creatine kinase, as you may know, leaks out of muscle fibers when they're injured. So when the membrane becomes upset, then what happens is proteins leak out of those muscle fiber tubes into circulation, and you can measure them. Now, creatine kinase isn't the only thing that leaks out. There's lots of other proteins too, some that you may have heard of and some that you may not. And I have 3 examples here: myoglobin, fast troponin, and creatine kinase. And one of the things that we'll be telling you about is fast troponin, which is a protein that leaks out of just of fast fibers. So there's actually a different troponin for cardiac, slow and fast muscle. And you can actually use it to say, well, which fibers are leaking and how is your intervention changing that leak? So we'll show you a tiny bit of data associated with that. This is how creatine kinase and fast troponin look if you have Duchenne, Becker, or if you're a healthy individual. And this is something you're probably familiar with, that Duchenne tends to have very high CK, Becker in the middle, and then healthy folks, it can spike up and down, but it's generally low. See on the left-hand side, that's also the case for fast troponin. And CK and fast troponin tend to go together, such that if your CK is high, your fast troponin is high. Now, this is just one little preclinical thing before we hand it off to Joanne to talk to you about the clinical studies. Probably the most complete model of Duchenne are these dogs with the disease. So these are golden retrievers that were identified that don't have dystrophin. And in many ways, they recapitulate many of the features of Duchenne. So the dogs get weakness, the muscles get fibrosis. And we decided to try a short study with 5506 to see how the compound would work in these dogs. And we actually used dogs that were older and had stabilized disease. So we didn't do a prevention study, we just did a real life, let's treat a dog that's weak. And what we saw is 2 things with this short-term study. On the left-hand side, we saw a decrease in CK. As I mentioned, CK leaks out of muscle fibers, and we decreased how much CK was leaking out. You can see that on the left. And on the right-hand side, we actually used a collar-bound activity monitor and measured how much those dogs were active during the day. And they're actually more active. So even though CK was decreasing, their activity was increasing. So we changed that balance between activity and injury. And it's exactly what we're hoping to see in folks in clinical trials. So I'll now hand it over to Joanne, and she'll tell you about what we see as the spectrum of disease, and then how far we've got in the clinic so far.
I need to turn on my, there we go. So we're looking at both Becker and Duchenne. And, you may ask, well, those are different diseases. How can the same compound do that? And as you know, in Duchenne, there's no dystrophin. In Becker, there is some dystrophin, but it doesn't work right. So it basically, in either case, you have the same problem that Alan just showed you in those muscle fibers, that they are subjected to stress and damage when the muscles contract. So that's what we are trying to prevent. And we think that EDG-5506 can prevent that in both diseases. So we're developing the drug both in Becker and in Duchenne. So I'll tell you a little bit more about the trials in both those diseases later on. So we started in humans, and basically the way that drug development starts typically is to start in unaffected adults. We also started in adults with Becker because we could see the biomarkers that Alan showed you, and we could see if they changed with EDG-5506. So what did we find out the first time we went into people? We first want to know a lot about safety. You guys want to know a lot about safety. And what are the side effects? Are there any problems with labs and things like that? And so what we saw early on and in continued studies is that EDG has been well tolerated. The most common side effect is that happens early on is some patients have some drowsiness. They may feel odd. They feel what's called dizziness. And that usually happens within the first few days of taking the drug but then it disappears. So it seems like the patients get used to that and it doesn't bother them. They don't notice anything anymore. It's given as an oral tablet once a day. We actually give it at night, and it can be taken with or without food, so that's convenient. And one of the things that we were able to look at when we looked at this for 2 weeks in what's called a phase 1 unit, where people are inpatient in we get to, we draw blood frequently. We also did muscle biopsies. And what we found was that EDG-5506 is very concentrated in muscle. And that tells us that because the drug is 100 times more concentrated in muscle in unaffected adults than it is in the circulating blood, it tells us that it's getting to where it needs to be. It's going to the protein that we are targeting. So that was very encouraging to see. And as a side note, I would say that the patients with Becker who came in and did this really got a gold star, because this is not great accommodations. So it really was asking a great deal of them to come and share their time and effort with us. So after that, actually, those patients with Becker continued on in an open label study as well. And we looked at the biomarkers, because we know biomarkers can change quickly. We saw them change after 2 weeks, and now we're looking at them up to 6 months in this open label study where everybody knows they're getting the EDG-5506. So there's 12 patients here. And we saw creatine kinase fall 39%. We saw the target of the drug, the fast muscle fibers, we saw that biomarker drop by 75%. So again, we're seeing that these biomarkers of muscle damage that tell us whether the muscle is being damaged are down quite a bit, which is really encouraging. And we saw that early on in a few weeks. We see it continuing out to 6 months. So that gives us confidence in also starting other clinical trials that I'll tell you a little bit more about. Now, the other thing we've seen as we are getting to the 6-month point, and we're continuing on as well, is we look at functional assessments. And as you know, in trials in Duchenne and Becker tend to be a year plus, because it takes a while for changes to happen in the muscle that are reflected in function. We're looking at the North Star ambulatory assessment. Many of you will have seen that in clinic. 17 different measures that assess whether or not boys and men can do certain functions, or whether or not they need to compensate because of weakness in doing those functions. And what we saw, the maximum score is 34. What we saw is that compared to natural history studies where people typically went down by 1.4 points per year, and you'd expect that would continue, we saw the North Star improving somewhat. And that gray area tells us statistically that's where the likelihood of the real effect is. So it's promising. Again, it's 12 patients, open label. So that doesn't mean that we know the drug works, but it's really encouraging for moving to the next studies. We also looked at an adult measure, the North Star assessment of limb-girdle type muscular dystrophies, and that also improved, although we don't have natural history data on that as well. So this was good. So we have now started, all of these studies are ongoing. The ARCH study is the study that I just showed you results from. We also have started the Canyon study. And now that's a study in Becker in adolescents age 12 and up and adult males. That's enrolling at a number of sites in the United States. It's also going to be enrolling in the UK and the Netherlands as well. And that is a 1-year study to look at function to look at biomarkers and most importantly, to look at safety in patients with Becker. The additional trial that we have just started within the past few weeks is called the LYNX trial. And I think Abby may have mentioned we're in Colorado, so that's why all of these trials have kind of Colorado Western kind of names. But the Duchenne study is in boys 4 to 9 who are ambulatory, and I'm going to tell you more about that on the next slides. This study is also a year long. It's a 2-part study. And the major objective, this is the first time we will be looking in young boys. So we want to look at safety. We want to look at biomarkers and identify the doses for a larger phase 3 study. So we're going to be looking at boys 4 to 9 who have Duchenne, who are on a stable dose of corticosteroids. That would be 6 months on a stable dose of steroids. They can also be on an antisense exon skipping drug that is at a stable dose for a year. So including those. And that kind of goes to, we're thinking that this kind of medication could work not only by itself, but also in combination with other treatments. We plan to enroll 27 boys across the United States, and some site, there's an Atlanta site that's open. The other sites we are getting on board as quickly as we can. And there'll be a 3-month period at the beginning of the study that's placebo controlled. And that will give us information about safety. We know the biomarkers actually change very quickly. And that will give us a good idea about what kind of a dose to use in the phase 3 study. That's followed by an open label period where everyone will get drug during that period. And that will allow us to see the changes in function over a year period in these boys. And as always, we will be then planning a open label extension beyond that for boys that have completed the 1-year study. There will be about 10 site visits. They're mostly concentrated at the beginning, about every 2 to 4 weeks for the first 3 months. And then we space them out so that at the end, it's about every 3 months. We do that because the first time we're looking in younger boys, we want to really monitor them for safety, not because we think anything bad is going to happen, but because we just don't know, the first time in boys. So we want to watch them carefully with, by the principal investigator. We have also, though, incorporated some elements to look at things and how things are happening at home. And I'll tell you more about that to try to minimize the number of visits. There are functional measures that we'll be looking at. We're not going to be looking at muscle biopsy. And we are going to be looking at the heart with an echo. We'll be looking at a DEXA scan, which tells us about bone density, tells us more about muscle mass as well, and lung function tests as well. And then the standard kind of functional measures, the North Star, muscle strength testing, and time function tests. So this speaks to what we're also exploring to try to minimize visits. And on the left is a device that we are using for the first time in this study in children. We've looked at it in adults. And the idea is to be able to draw blood at home in a very minimally invasive way. That device sits on there's an adhesive, the parent puts it on the upper arm and pushes that red button. And you've seen things like glucose monitors, which have a tiny needle. The reason that it's on the upper arm is because that is a less sensitive part. It's much less sensitive than, say, a fingertip, which hurts when you prick it. So I've done it, others have done it. It doesn't hurt, but we can't promise it doesn't hurt, but it's really quite minimal compared to a blood draw for example. So we're using that, and then the parent would put that in a box and send it back that we supply to gather more information on things like blood levels of the drug. We're also looking at at-home activity monitor with these devices that kind of look like Apple Watches, and they are worn at different time points during the study, during daylight, and they're relatively small on the wrist or the ankle. As we progress in the clinical development program, we'll be looking at other age ranges and ambulation status. We very clearly know that we have to think about how are we going to bring this into the non-ambulatory population, both in Becker and in Duchenne. So that's something that we are thinking about actively. The Duchenne study is in the United States, but when we get to phase 3, and this is designed to get us to a phase 3 study for registration, we'll be looking beyond the United States at a number of other countries. And we always think about this in terms of, well, what's the standard of care as new medications are approved? We are including exon skipping. That's an important addition to current treatment, and will be allowing, as I mentioned, boys who are on exon skipping to participate. So that's, we are thinking, as Abby mentioned at the beginning, on how we can reduce the burden, because as much as we try, we know that this is a burden in trials. I mean, you've got enough to do, and to add this on, we realize, but it's really important in order to bring things forward and to make new, and hopefully make new therapies available. As much as possible, what we are doing is providing concierge services for travel, so people don't have to pay up front. They arrange things to ease the whole burden of participation. So, help with siblings in terms of travel if that's needed to the site, or childcare expenses. And as I mentioned, trying to do things at home as much as possible to minimize the burden. So kind of to summarize, this is a agent that is taken orally. And the idea is really to preserve and to improve function in the muscle in both Becker and Duchenne patients, regardless of mutation. The goal is really to protect the most susceptible muscle fibers, and as Alan showed you, to prevent damage and allow that muscle to regenerate as well. We think that this can be used alone, and we'll be looking at it in combination with other therapies, and really to stop the damage where it begins, where muscle contraction damages the muscle, releases these biomarkers. And if we can target that initial damage, then we think that we can have a big impact on disease progression. So with that, I want to thank you. It's always a pleasure to be here with the Jett Foundation. They do so much good stuff. It's great to see always. And we'd be happy to answer questions if Alexa has some.
Thank you, Abby, Joanne, and Alan. We really appreciate you. At this time, we don't have any questions. Abby did answer one in the Q&A already, but if anyone does have any other questions, please feel free to ask them. I think someone's asking about open and nonsense mutation.
Yeah. So in theory, this should work on top of a nonsense mutation as well, that is providing some dystrophin. We know that atalurin, the levels have to be tightly controlled, with the dosing. So we would not want to, we would want to do a drug interaction study first before doing that. So on top, it should work in boys that have a stop codon. The only issue is on top of atalurin, but in general, that's not, and that's an investigational agent, and Typically, as in our studies, you can't be on another investigational agent during the study.
It does bring up the, something that's close to our hearts though, Joanne, which is combinations with other stuff. And 5506 is quite different from anything else that's been tried. So as a structural fix, we're kind of immune to what kind of mutation you have, whether you have a stop, a nonsense, a large deletion, whatever it is, it doesn't really matter. And as such, the novelty of the therapy speaks well, I think, to potential combinations, right? So in the Duchenne studies, they'll be stabilized on steroids, right? We've done some preclinical work to look at combinations with gene therapy, and that's a great interest for us on the microdis. And similarly, other developing therapies should be okay to work with this too. So that's a good opportunity, I think.
Yeah. In the Duchenne study, there'll be 27 enrolled. And of course, when we have those data, then we'll be moving on to hopefully to a phase 3. And that would be a larger study as well. In the Becker study, there's going to be 66 in all.
All right, if anybody has any other questions, they have their email up there that you can ask, but you can also always email me, alexa@jettfoundation.org, and I will make sure that it gets to the Edgewise team. Thank you again for being here. Like I said, this was recorded, so if you missed anything, you want to rewatch, you want to send it to someone who you might find would benefit from it, please, go to our YouTube in a day or two, and it should be up there. For all of you that are registered, you'll also get an email with a survey and the recording link as well. We hope to see you again for our community webinar series in the future. We will have 2 more this year, so go ahead and check those out, and then we will start back up with our Rare Disease Day in February of 2023. So thank you guys so much. And again, Abby, Joanne, and Alan, thank you guys so much for being here and sharing this information. I think it's very, very exciting, and we're very excited for you guys in the community. So thank you guys for being here and sharing this. We appreciate it.
And I hope everybody has a wonderful holiday too next week.
Yeah, I might.
Oh, yeah.
Coming up, it's crazy. I always say this, I've been saying this on the last 3 webinars. I don't know how we're at the end of November already. It's crazy, but yes, absolutely. We hope you all have a great holiday and a holiday season.
Thank you.
Thanks.
Have a good one. Thank
Everybody,
you guys.
bye.